All right, today we're going to be looking at chapter 16 over blood. And blood is the life-sustaining transport vehicle of the cardiovascular system. It's found within blood vessels, it flows through the heart, and we're going to see that it includes various functions. The functions are going to encompass transport, regulation, and protection. So let's look first at the transport functions. Of course, it's responsible for delivering oxygen and nutrients to body cells. One such nutrient would be glucose, and glucose and oxygen are commonly used by cells that can get oxygen in cellular respiration in order to create ATP. We're also going to see that blood is able to transport other substances besides oxygen. So we can also transport things like metabolic waste to the lungs and kidneys for elimination, or even transport hormones. We just got done, or you should have just gotten done, looking at chapter 15 over the endocrine system. We know that hormones are long-distance chemical messengers that travel via the blood to their target organs. There's also regulation functions for the blood. The regulation functions include maintaining body temperature by absorbing and distributing heat. Heat is oftentimes a byproduct of chemical reactions that occur within the human body, and that heat has to go somewhere, and it is absorbed oftentimes via the blood. We also see that the blood can maintain normal pH using buffers. There's an alkaline reserve of bicarbonate ions that we'll look in more detail as we go through this PowerPoint at and how it's going to help us maintain kind of a homeostatic range for the pH of blood. We also can see that maintaining adequate fluid volume in the circulatory system is possible, or we will see that is possible by the regulation functions blood does possess. There's also plenty of protection functions that are possible or can be carried out by the blood. So there's things like platelets, which are going to aid in the process of hemostasis in order to prevent blood loss. Plasma proteins and platelets are definitely involved in initiating clot formation. And then there's white blood cells, complement proteins, antibodies, plenty of agents of immunity that are carried in the blood and help prevent infection and are definitely a part of the immune system. So to kind of take a step back, now that we've talked about everything that blood can do, let's talk about histology. So if you think all the way back to chapter 4 in your lecture textbook, blood was considered a connective tissue, but it was an atypical connective tissue because it was the only fluid tissue in the body. It's a connective tissue because it had more extracellular matrix than cells, and it is a fact that plasma, the non-living fluid component of blood, is going to be the primary component in what we call whole blood. So the matrix or extracellular matrix for blood is blood plasma. So the cells are going to be the living component. As a group, we call all cells in whole blood formed elements. And these formed elements are going to be suspended in plasma. So the formed elements, the cells, are suspended in the extracellular matrix. And let's just list the types of cells that you can find in blood. Erythrocytes, leukocytes, and platelets. And of those three, leukocytes are the only ones that are complete cells. Erythrocytes are anucleate, they're missing their nucleus. And platelets are cell fragments. Some abbreviations for erythrocytes are RBC for red blood cell, leukocytes, WBC or white blood cell, and platelets are also known as thrombocytes. If we were to look at the composition of blood, we would look oftentimes at a spun tube of blood. So a tube of blood would be spun in a centrifuge, and because blood is a suspension, the components would filter out. And the erythrocytes or red blood cells are the heaviest, so they would settle out on the bottom. And that would make up about 45% of whole blood. That percentage of red blood cells that would filter to the bottom is the hematocrit, or percent of blood volume that is red blood cells for a sample. So the normal values of hematocrit in males can be slightly higher than that of females. White blood cells and platelets are going to make up a layer called the buffy coat, and less than 1% of a blood sample is generally made up of this buffy coat, which is white blood cells and platelets. And then the majority of spun blood is going to be made up of plasma, and plasma is the lightest and will be found at the top of a spun tube of blood, about 55% of the sample. So here we can see that in figure 16.1. We have plasma at the top, the buff coat in the middle, and erythrocytes at the bottom. So we have all the formed elements found within the buffy coat and the erythrocyte percentage, or hematocrit. Because erythrocytes are red blood cells, buffy coat contains the other two types of cells, leukocytes and platelets. So some physical characteristics about blood. Blood is sticky, it's opaque, it should not be clear, it has slightly a metallic taste. Its color can vary depending on its oxygenation. When it has high oxygen levels, it's generally more of a scarlet red versus when there's low oxygen levels to blood and it shows a darker red. The pH of blood is going to be slightly basic, so it's at about 7.35 to 7.45, just a little over neutral, and it makes up 8% of your body weight. And the average volume of blood in the human body is going to be slightly higher in males than in females, by about a liter, give or take. So now that we've kind of characterized the general functions of blood, what it's made up of, that it's a typical connective tissue, but something atypical about it, and that is that it's a fluid, let's now look at the components in more detail. So first and foremost, blood plasma. So blood plasma was kind of a straw-colored, sticky fluid. It's mostly water, but what's not water is the over 100 dissolved solutes. Solutes include things like nutrients, like glucose, gases, hormones, waste, protein, inorganic ions, just things dissolved in this watery environment, which is blood plasma. Plasma proteins are probably the most important thing here to look at, and they're the most abundant solute. They remain in the blood. They're never taken up by cells, and most of our plasma proteins are going to be produced by the liver. An important plasma protein to take note of is albumin. Albumin makes up 60% of plasma proteins. So not only is it an important plasma protein, it's an abundant plasma protein. The reason it's important is because not only does it function as a carrier of other molecules, such as a carrier of steroid hormones, it's also a blood buffer, but probably the most important function it has is maintaining oncotic or osmotic pressure. So it definitely contributes to maintaining kind of a pull on water and preventing all blood plasma loss or fluid loss to interstitial spaces and tissues. So here is just kind of an overview of the composition of plasma. You can see that its main constituent is water. There's a list of other solutes like certain electrolytes, sodium, phosphate, chlorate, sulfate, plenty of plasma proteins. Some of the plasma proteins you want to know other than albumin would be the globulins. Immunoglobulins are antibodies. You want to also note fibrinogen, which is going to be involved in creating a fibrin mesh, which is going to help us form a blood clot during the process of hemostasis. And then just be aware of general stuff you can also find in plasma, like, of course, the hormones we've talked about in the prior chapter, respiratory gases like oxygen and carbon dioxide, and plenty of nitrogenous waste, such as urea. And one major nutrient we'll talk about a lot is glucose. So now that we've talked about plasma, let's move on to the formed elements. The formed elements are red blood cells, also called erythrocytes, white blood cells, also called leukocytes, and platelets, which are also known as thrombocytes. And we've stated before that only white blood cells are complete cells because red blood cells are anucleate, and platelets are cell fragments of cells called megakaryocytes. So most of our formed elements survive in the bloodstream for only a short period of time, only a few days, because they're not complete cells. Even our immune cells only survive for a short period of time because a lot of them die fighting. Most blood cells do originate in the bone marrow, and from which they originate, they do not divide further after they're mature. So here's just an overview of the blood cells. This is figure 16.2a in your textbook, and we can see pointed out here the biconcave erythrocytes, the fragmented platelets, and the large leukocytes. There's actually various different types of leukocytes, and we kind of tell which is which based on its overall size, whether or not it has granules, what color are the granules, and the shape of the nucleus. So we see here a couple of different white blood cells, including monocytes, lymphocytes, neutrophils, and eosinophils. The other white blood cell in this image that is not labeled up here is also a neutrophil. Neutrophils are the most abundant white blood cell found in blood. And then we have these kind of donut or dish-shaped red pink cells. Those are our erythrocytes or red blood cells. And then these dots throughout are platelets or our thrombocytes that are involved in blood clotting. The white space is blood plasma. And we see that all these cells are suspended in that extracellular matrix. So next, let's move on and look more closely at our formed elements individually. So erythrocytes first. So erythrocytes are small. They contribute to gas transport. They primarily are known for transporting oxygen. They have a biconcave dish shape, so we'll see a picture of that. We know they're anucleate, so they don't have a nucleus, and essentially no organelles. They're basically sacs of hemoglobin. They're filled with hemoglobin, and hemoglobin is a protein that helps or functions in the transport of oxygen. It can also transport carbon dioxide, but mainly is known for its ability to transport or bind to oxygen reversibly. Red blood cell diameters are larger than some capillaries, so they have a membrane protein in them called spectrin. And so this membrane protein in the plasma membrane of red blood cells allows them to kind of bend and move and flex through these really small blood capillaries that are smaller than themselves. So the structural characteristics, as we look further at red blood cells, are going to display a great example of complementarity of structure and function. And if you forgot what that means, we could go all the way back to chapter 1 in your lecture textbook. But essentially, complementarity of structure and function is that structure matches function and vice versa, that they go together, that they complement each other, that they work together and they make sense when we analyze something both anatomically and physically. The three features that make for an efficient gas transport system when we're looking at the complementarity of structure and function that is red blood cells are the fact that red blood cells have a biconcave shape. It offers a huge surface area relative to volume for gas exchange. What this means is this biconcave shape makes it where that no place in the cytoplasm that contains hemoglobin, which usually holds oxygen, is too far from the plasma membrane. So it means that red blood cells are great at reversibly binding and unbinding to oxygen, not just because they contain hemoglobin, which can do so, but because these proteins are always going to be relatively close to the plasma membrane, be able to function in gas exchange. Hemoglobin also makes up 97% of the cell volume, and that's not counting water, but that means that red blood cells are so dedicated to their job of carrying respiratory gases that they're made up pretty much entirely of this protein that does so. And also, red blood cells have no mitochondria, so they're not able to make ATP, but this also means that they're uninterested in using their own oxygen supply. So ATP production for red blood cells is anaerobic, so they're not consuming the oxygen they transport. Here is the biconcave shape for erythrocytes or red blood cells. So both terms mean the same thing. So this is a side view or kind of a cross section. And we can see that on both sides, the middle of the red blood cell kind of pinches in or is concave. Kind of looks like an incomplete donut. The outsides are going to be kind of bulbous and expanded. It kind of looks like a tire that wasn't completed. So let's move on and look at the function of erythrocytes in more detail. We know that they bind oxygen. We know that red blood cells are able to bind oxygen because they contain hemoglobin. That's been discussed kind of in depth, but let's just look at it in more detail. So red blood cells are dedicated to respiratory gas transport. And we're going to focus a lot on oxygen, but what respiratory gas transport implies is that red blood cells can transport both carbon dioxide and oxygen. Those are our two respiratory gases. But hemoglobin is known best for its ability to bind reversibly with oxygen, and the amount of hemoglobin in the red blood cells of males tends to be just slightly higher than that of females. And hemoglobin consists of a couple of components. First and foremost, there's heme. These heme groups are pigments which contain central iron atoms. There are 4 heme groups in one hemoglobin molecule. Every heme group which contains a central iron atom is found in one of 4 protein molecules, or polypeptide chains. There's actually 2 alpha and 2 beta chains, and along with our chains, the heme and the iron make up the components that we're going to see in hemoglobin. Now, the reason for the name hemoglobin is first the heme groups, that's where we're getting the heme, but globin comes from the polypeptide chains, which are known as globin chains. So let's look at the structure of hemoglobin. This is figure 16.4a in your textbook. So we have 2 beta globin chains, 2 alpha globin chains, and then we have a heme group in the center of each globin chain. So since we have 4 globin chains, we have 4 heme groups, and in the center of each heme group is an iron atom. So here is our iron-containing heme pigment, and it's to this iron molecule that oxygen would bind. So if you're ever asked where does oxygen bind on hemoglobin, the answer is to the central iron of a heme group. And if there's 4 heme groups, each with a central iron in hemoglobin, the answer to how many oxygen molecules can hemoglobin bind to, 1 hemoglobin molecule, that would be 4. So each hemoglobin molecule can transport 4 oxygen molecules, and each red blood cell contains 250 million hemoglobin molecules. So what that means is just 1 red blood cell can transport about 1 billion oxygen molecules. We see oxygen loading in the lungs. It makes sense that our blood is going to get oxygen loaded into it or from the lungs at our respiratory center, which includes our 2 lungs and the pulmonary capillaries that serve them. And this would produce oxyhemoglobin. So when oxygen is loaded into the lungs, we produce oxyhemoglobin, which has a more kind of bright ruby red color. Oxygen unloading occurs in the tissues, so that means that the blood is going to give up its oxygen and become deoxyhemoglobin. Now, we say the blood's going to become deoxyhemoglobin, or we're going to produce oxyhemoglobin when the blood gets oxygen. But what's really happening is oxygen is going to be bound to hemoglobin to produce oxyhemoglobin. For the unloading, oxygen is going to unbind from hemoglobin in order to produce deoxyhemoglobin, or what's called reduced hemoglobin, which has a much darker red color. And don't forget carbon dioxide, or CO2. Its loading occurs in the tissues, and actually about 20% of carbon dioxide can bind or does bind to hemoglobin. And when carbon dioxide is bound to hemoglobin, that produces carbaminohemoglobin. Carbaminohemoglobin is just a fancy way to say that carbon dioxide is bound to a hemoglobin molecule. But unlike oxygen, carbon dioxide binds to the globin chains of hemoglobin. So theoretically, oxygen and carbon dioxide can be bound to hemoglobin at the same time. However, carbon dioxide binding to hemoglobin is going to increase the affinity of oxygen to unbind from hemoglobin. This is going to make sure that oxygen unloading occurs in tissues, whereas CO2 loading to hemoglobin oftentimes only occurs in tissues, so that we have this reversal of respiratory gas transport. Let's move on and talk about the production of erythrocytes, not just what they look like and how they work and their function, but let's talk about hematopoiesis. So hemato, or hemat, is going to be a root word that means blood. Poiesis means generation or production. So this word means the generation or production of blood. So more literally, it means the formation of all blood cells. And that occurs in red bone marrow. Red bone marrow tends to be composed of reticular connective tissue and tends to have blood sinusoids, which are a type of capillary with really big holes called sinusoids. So in adults, you'd find red bone marrow in the axial skeleton, the pelvic girdle, and in the proximal epiphyses of bones like the humerus and the femur. And in the red bone marrow, you would have these stem cells. These stem cells are called hematopoietic stem cells, or sometimes hemocytoblasts. Hematopoietic just means, again, blood generating. So we're keeping kind of everything very literal, blood generating stem cell. Even with hemocytoblasts, that means blood cell that produces, because blasts produce something. If you think back to AMP1, you've probably heard of an osteoblast or a chondroblast. They produced a matrix. And in this case, what we're producing is sort of a matrix in this kind of bone marrow environment, but we're actually producing what's going to be the formed elements of whole blood. So hematopoietic stem cells are going to be stem cells that give rise to all formed elements. That includes white blood cells, red blood cells, and platelets. Definitely get comfortable listing your formed elements whenever called for. There's going to be hormones and growth factors that are definitely pushing the cells toward becoming a specific type of cell, a red blood cell, a white blood cell, or a platelet. And once the cell commits to becoming a certain type of formed element, it cannot change its mind. So there is going to be a committed stage. And once the new blood cells are mature or almost mature, they are able to leave via the blood sinusoids and enter regular circulation. So here are the basic stages of erythropoiesis. Erythropoiesis is a term that's a little bit different than its other term, hematopoiesis. Hematopoiesis is a more general term. It's the formation of all blood cells, whereas erythropoiesis is more specific. So red poiesis, generating or producing. So this is red producing, or the process or formation of red blood cells. This process is looking specifically at the formation or production of red blood cells. So there's a couple of stages of transformation. Of course, we're going to start with our hematopoietic stem cell, which then will transform into a myeloid stem cell, then a proerythroblast, and then we'll go through a couple of different stages of erythroblasts. That's after the proerythroblast. Those include the basophilic, polychromatic, and orthochromatic erythroblasts. And then finally, the developing erythrocytes will become reticulocytes. Reticulocytes are considered immature red blood cells, but they're almost a mature red blood cell. They're almost a mature erythrocyte. But reticulocytes still contain a small amount of ribosome. And so they have to get rid of that ribosome, make sure they spit out their nucleus. It may have begun degrading in step 6 when it's an orthochromatic erythroblast. And that happens about 2 days after reticulocytes enter regular circulation. So reticulocytes are actually what leaves the red bone marrow and enters regular circulation. And then in 2 days, that reticulocyte will become a mature erythrocyte. So you can actually count reticulocytes to indicate or get a measure as to the rate of red blood cell production. So I really like figure 16.5. We can get a general idea of the erythropoiesis process or formation of red blood cells. Make sure you know what the stem cell is, the hematopoietic stem cell or hemocytoblast. The pro stage here is the committed cell. There's no going back once the cell becomes a proerythroblast. What's missing here is the derivation of the hematopoietic stem cell into the myeloid stem cell. That's just a small thing that's left out. And then the developmental pathway that includes 3 erythroblast phases. At the end of the last erythroblast phase, we have the ejection of the nucleus. Sometimes finishes up during the reticulocyte phase, but mainly we're just getting rid of ribosomes during that reticulocyte phase. The reason we have all those ribosomes in the reticulocyte phase is the immature red blood cell is still generating all that hemoglobin. So by the time it becomes a mature erythrocyte or a mature red blood cell, it should have enough hemoglobin that it can be considered, again, that great respiratory gas transporter, basically a sack of hemoglobin ready to just transport substances like oxygen and carbon dioxide. So let's look at some of the regulation and requirements for that process. It can't just happen whenever it wants to unless it has nutrients, unless it has supplies. And first and foremost, we just want to establish kind of a couple terms and things that occur. So first, too few red blood cells leads to tissue hypoxia. Tissue hypoxia is a condition in which tissues are not getting the oxygen they need via blood supply. So that's bad, but also too many red blood cells is bad because that increases blood viscosity or blood thickness. Kind of makes your blood more like a sludge, and that can lead to blood clotting and strokes. And we really want to keep the amount of red blood cells being generated on par with the amount of red blood cells being destroyed. So with about 2 million red blood cells being made per second, we want to destroy about that many. A lot of them are broken down in the spleen, which is called the red blood cell graveyard. So again, we're balancing red blood cell production and destruction. And our ability to do that and do that in order to maintain a homeostatic balance depends on hormonal controls and certain dietary requirements. One hormone is erythropoietin, which showed up briefly in the Chapter 15 PowerPoint. Erythropoietin is abbreviated EPO, and it's just a hormone that stimulates the formation of red blood cells. Now, erythropoietin is secreted by the kidneys, and is released by the kidneys, some from the liver, in response to hypoxia, or low oxygen levels. At low oxygen levels, there is an oxygen-sensitive enzyme called hypoxia-inducible factor that cannot be degraded. Now, it cannot be degraded in kidneys. When this HIF or this factor builds up because it can't be degraded when there's low oxygen levels, that's going to trigger the synthesis of EPO. So HIF accumulation is the trigger for EPO production from the kidneys. But in general, there's always a small amount of EPO in the blood to maintain the basal rate. But if we want to see this extra production, this production that's going to increase or cause a stimulation that forms red blood cells, we would have to see low oxygen levels be the primary trigger. So causes of hypoxia include decreased red blood cell numbers, maybe due to hemorrhaging, some type of injury that's causing massive bleeding, insufficient hemoglobin per red blood cell, iron deficiency anemia is a great example, or reduced availability of oxygen, such as that occurs with a blood clot, or when someone goes up to high altitudes all of a sudden, or has lung problems, such as pneumonia. The hormonal control is going to play a role in, again, red blood cell production. Because if there's too many erythrocytes or high oxygen levels in the blood, we're going to inhibit EPO production. So there is that negative feedback. There is a check and balances when it comes to the production of EPO from the kidneys. Because when EPO is produced, it causes erythrocytes to mature faster. It causes reticulocytes to be dumped into circulation much faster. And because testosterone enhances EPO production, that accounts for the high red blood cell counts in males compared to females. So here, let's look at figure 16.6. Definitely a figure that I recommend studying. It's looking at EPO, or the EPO mechanism for regulating erythropoiesis. So we have first kind of the normal blood oxygen levels. Then we're going to have something that sets us off balance. The stimulus that sets us off balance or disrupts homeostasis is going to be hypoxia. That is the stimulus that causes the kidneys to release erythropoietin. Notice that hypoxia, however, does not directly stimulate the bone marrow. It's renin that acts on the bone marrow to cause it's EPO from the kidneys that acts on the bone marrow that causes it to create or produce more red blood cells. So in the last chapter, we talked about renins. I still have that on the brain. But it's hypoxia that causes the production of EPO from the kidneys that acts on the bone marrow that causes the production of red blood cells that therefore increases the oxygen carrying capacity of the blood. So there are some questions that are kind of trick questions that you may see if you're looking at practice exams or using Mastering A&P, where they ask which one of the following directly stimulates bone marrow to produce more red blood cells. And I've seen a lot of students choose hypoxia as the direct stimulus for bone marrow. But hypoxia is a direct stimulus for EPO production, and it's EPO production which is the direct stimulus for red bone marrow erythropoiesis. So just make sure that you kind of understand this pathway and you don't skip a step. It's EPO that acts on the bone marrow. Let's look at a homeostatic imbalance. So this is imbalance 16.1, and we're talking about some athletes who can abuse or do abuse artificial EPO. So artificial EPO is an artificial hormone, and if you use it, you can increase your hematocrit or amount of red blood cells in your blood. And that would allow an athlete to increase their stamina and performance. They would have more blood cells, more oxygen carrying capacity, and sounds pretty good as far as athletes are concerned. But there's dangerous consequences. You do not want to increase your hematocrit too much. If you increase it from 45% up to even 65% by abusing artificial EPO, and then you add on to that dehydration, which is commonly associated with kind of the after effects of any type of physical performance, then you're going to put yourself at risk for the blood becoming very thick, very sludge-like, possible blood clotting, possibly a cerebral embolism or pulmonary embolism, stroke, or even heart failure. The dietary requirements for erythropoiesis include pretty much the stuff to build hemoglobin, things like amino acids to build the globin chains, lipids, carbohydrates, iron available from the diet, iron because there is a central iron atom in each heme group. And most of iron is actually found in hemoglobin. That's kind of where it stays. The rest is stored in the liver, the spleen, and the bone marrow. And we want to store it or have it in hemoglobin because free iron ions are toxic. So we want to bind it to something that can transport it or something that can store it. Things like ferritin and hemosiderin store free iron ions in cells, so they're no longer free. And then we can transport iron ions in the blood if we bind the iron ions to transferrin. We also want to make sure that the body has vitamin B12. Vitamin B12 and folic acid are both necessary for the DNA synthesis of red blood cells. Red blood cells divide very rapidly, and that rapid division requires vitamin B12. Erythrocytes do not have a nucleus once they mature, so their lifespan's not going to be very long, about 100 to 120 days. Again, they're anucleate, they don't have ribosomes, they don't have most organelles, they can't synthesize new proteins or aerobically make ATP, so they're slower at making energy. They're not going to grow, they're not going to divide, so eventually they do become fragile. Spectrin starts to degrade, hemoglobin starts to break apart, and the old red blood cells can get trapped in smaller circulatory channels, and this occurs especially in the spleen. Then macrophages in the spleen engulf and break down the dying red blood cells. Macrophages are white blood cells that are great phagocytes. So red blood cell breakdown includes breaking down the components like heme, iron, and globin, separating them, binding iron to storage molecules or transport molecules, breaking down heme, which is a pigment, into bilirubin, then urobilin, and then stercobilin. Stercobilin is going to be a brown pigment that leaves the body in feces. And then we'll really metabolize or break down globin into its subsequent amino acids. And those are just released into circulation to be used to build other proteins. So that process is listed here. So we have step 1 at the top, where low oxygen levels in the blood stimulate the kidneys to produce erythropoietin. Step 2, erythropoietin levels rise in the blood. Step 3, erythropoietin and necessary raw materials in the blood promote erythropoiesis in red bone marrow. Step 4, new erythrocytes enter the bloodstream, and erythrocyte function for about 120 days. And that would lead us to step 5, where our aged or damaged red blood cells can be engulfed by macrophages of the spleen, sometimes the liver and bone marrow. And the hemoglobin is going to be broken down into heme and globin. And then we saw that the globin is broken down to its subsequent amino acids, which can then be released into circulation. And they would be used as needed. The iron has to be stored. It'll be transferred via transferrin. The liver releases it to the blood as needed for erythropoiesis. And the rest of the heme is going to be broken down to bilirubin that is picked up by the liver. The liver secretes bilirubin in bile into the intestine, where bacteria metabolize it to stercobilin. Stercobilin is excreted from the body in feces. And step 6, raw materials are made available in the blood for erythrocyte synthesis. So food nutrients such as amino acids, iron, B12, folic acid, they would all be absorbed from the intestine and enter the blood. So I know that was long, but that's what we're seeing in figure 16.7, which is the life cycle of the red blood cell. So this is a great study image. It puts together all that information we were looking at for the function and the regulation and kind of the process of erythropoiesis and the triggers for what causes the production of EPO that then upregulates the production of red blood cells, and so on and so forth, all the way to its degradation.